Welcome to another episode. So far we've talked about fats and carbohydrates as two of the three macronutrients. Today we're going to be talking about protein. Now protein is really important. Proteins are involved in more types of cell functions than any other type of molecule. They function as enzymes to catalyze reactions. They're antibodies in our immune systems. They facilitate movement, such as peristalsis, which moves food through our digestive tract. They're involved in cardiac functions, running, walking, all those things. They're used as signaling molecules, such as insulin instructs our cells to store energy when too much glucose is in our bloodstreams. They're also involved as transport mechanisms to get nutrients across our cell membranes, both into and out of our cells. Now, because proteins are made of amino acids, there were a number of experiments done in the 1950s to determine sort of how these things were formed. So in 1953, a guy named Stanley Miller conducted an experiment. He took a couple of glass flasks and connected them with some glass tubing. And in one of the glass flasks, he had just regular water. And in the other glass flask, he had electrodes. So the water was boiled, and the vapor that came from boiling that water would go through the tube and over to the other flask. Now, this flask had not only the electrodes into it, but it also had some gases. Those gases were ammonia, methane, and hydrogen. Now, these three gases, ammonia, NH3, methane, CH4, and hydrogen, H2, made up three different components, atoms, that were very important in trying to determine if a secondary type of chemical could be created. Now, these three atoms were hydrogen, nitrogen, and carbon. Miller wanted to find out if some of the building blocks for amino acids could be created with an atmosphere that he believed existed in the ancient Earth. So once again, he boiled the water in the first flask. The vapor went from that flask through a little pipette or whatever glass tube to the second flask that had these gases. And as these gases took up this flask and the moisture came in, he was also charging these electrodes, causing sparks to simulate lightning. And then the moisture would condense and go back through a tube into this fake ocean flask again, and then boil again and go through this cycle. So this continued, and after a full day of doing this boiling and the electric charging those simulated lightning bolts, he noticed that the ocean water began to turn pink. And after a week of doing this, the ocean water had turned red, which is really quite amazing. So after this week of boiling and shocking this stuff, this dark red material ended up containing some of the things that he was wondering if it would actually occur. Things like hydrogen cyanide and formaldehyde, as well as some larger molecules of amino acids. Now, all of these are considered building blocks for amino acids and proteins. Miller's work was also criticized by those who said that taking volcanic gases and putting them into a glass flask and exposing them to radiation such as sunlight would also create the precursors to amino acids. In addition, a number of amino acids could also be formed in experiments that mimic volcanoes in deep oceans. Amino acids are also found to be in meteorites, so they can be formed in a lot of different environments. That's kind of the history of how it was decided that amino acids could be formed in the environments that were present on the ancient Earth. So amino acids, what are they? So there are 20 amino acids. And of them, there are 9 essential amino acids that our bodies cannot synthesize. We need to eat those to get them. Now, the structure of amino acids are all relatively similar, except when it comes to this section called a side chain. So first of all, they're all surrounding a carbon atom. And out of the top of the carbon atom, there is a hydrogen atom. And off to one side is a carboxyl group, and the other side is an amino group. Out of the bottom, then, is a group, an R group, that consists of a number of side chains, these 20 different side chains that determine which amino acid is formed. Now, this list includes all 20, but they're not in any specific order. They're basically just in alphabetical order. The biggest difference is the side chains and their affinity to water. In other words, some are more hydrophilic, they like water, and some are more hydrophobic, they don't like water. So they're either soluble or not soluble in water. And just that hydroaffinity, that hydrophilic or hydrophobic affinity, really drives a lot of the shape and the function of proteins. Proteins then are just chains of amino acids strung together. In general, a peptide is any string of amino acids that is less than 50 amino acids long, and a protein or a polypeptide is any string of amino acids that is 50 amino acids or greater. So each amino acid is considered a monomer, and when they are put together, they are polymers. So when they are put together, they are polymerized. And these bonds that bind them together are called peptide bonds. To go a little bit further down into the rabbit hole, when these amino acids are joined together, they are then considered residues. When they're singular, they are amino acids, but when they're joined together, they are amino acid residues. So a string of amino acid is a number of residues. Now, the structure of a protein really determines its function. So within the structure of a protein, there are 4 levels to this organization. It's kind of a hierarchy of structural formations. The first one, the primary structure, is just that string of amino acids strung together. That's the primary structure. And that primary structure can consist of those 20 amino acids in any order at pretty much any length. So it's really limitless on how many proteins can be formed. And because of their charge and also their affinity to water, the way these amino acids are joined together really determines the function and the organization, the structure, the final structure of the protein itself. So the secondary structure is determined by how these amino acids can join together with hydrogen bonds within them. There are twisted or helical structures. There are also folded or pleated structures. And these different types of structures, these secondary structures in a long protein, can be in the same protein at the same time. A protein can contain helical structures and the folded, pleated structures all in one protein. The third type of structure really takes this protein and just folds it and mutilates it, kind of. So the way these amino acids can react with each other, and these helixes and these folded, pleated portions kind of get close to each other, and if they're forming in a liquid, then the parts that are hydrophobic want to stay on the inside. The ones that are hydrophilic don't mind staying on the outside. It starts to form that way. There are other kinds of bonds that occur within it. So basically, the third structure is even more twisting and folding of this protein. And the fourth structure, the quaternary structure, is basically taking a couple of different peptides and joining them together. So you have 2 or more polypeptides. All right, so far we've talked about amino acids and how an amino acid is formed. It is a carbon that has a number of things around it on its 4 arms. So there's a hydrogen, there's a carboxyl group, and an amino group. Fourth bond is bound to this R group, or this side chain. The side chain is what determines the form and function of this amino acid. And then these amino acids, once they're bound together with these peptide bonds to form either a polypeptide protein or a peptide, those amino acids then help to determine that peptide or that protein's final structure and its function. When we eat proteins, our digestive tract mutilates these proteins and breaks them up into amino acids. None of this stuff survives our gut. And these amino acids then are distributed throughout our bodies and used to synthesize proteins. And protein synthesis is done at the cellular level. Within each one of our cells is a nucleus, and within the nucleus there is DNA. That DNA determines what messages are sent to different organelles in our cells to tell it what sort of proteins to create. So the first part of creating a protein is done within the nucleus, when certain sections of the DNA are identified. Now, amazingly enough, there are ways that our bodies know which sections are determined to be the right place to look in the DNA to find this recipe for what the protein should look like. And because we're so incredibly complicated, it takes a copy of that section of DNA, that gene, but that section isn't really all there is. In fact, there are many sections of this section that is transcribed, sort of like if you were taking notes from someone, you'd be transcribing those notes. You'd be transcribing what they were saying. That section of transcribed RNA from the DNA includes sections that are used for the final processing and sections that are not used. So the sections that are not used are called introns because they are intervening sections. There are sections that are used that are called exons because they are going to be expressed in the final copy of the RNA. Even though this is an incredibly complicated thing that eukaryotic cells like ours do, it ends up working out because we're alive. So anyway, these introns are removed and we finally end up with a section of RNA, specifically an mRNA, messenger RNA, that is sent out of the nucleus then into the cytosol, into the cell, where this section of messenger RNA, mRNA, is taken up by an organelle called a ribosome. Once this ribosome attaches to the mRNA, the next part of the transcription and translation portion of creating a protein begins. We've already gone through the transcription portion, so now we're going to translate it. Think of it sort of like if you were at that meeting and you were transcribing the notes, but the notes were in Spanish. Now you want to give those notes to someone else, but before they can understand what's being said, you have to translate that. So that's basically what is being done. First, the RNA is being transcribed from the DNA to create a copy, and then that copy is translated by the ribosome to create a protein. That mRNA is translated through the ribosome to create the protein. Now the mRNA has little triplets, 3 nucleotides that are read by the ribosome. So the ribosome reads these triplets, these codons, to create an amino acid. So 3 nucleotides within the mRNA, little sections, each 3 triplets, each triplet of nucleotides is considered a codon. Each one of these codons then codes for a specific amino acid. So as the mRNA passes through the ribosome, or in one end goes the mRNA with the codons, and out the other end comes amino acids linked together. And as that process continues, the mRNA goes through the ribosome and creates a protein. Once the protein is formed, it needs to develop its final structure based on those 4 structures we talked about first, the primary, secondary, tertiary, and quaternary. The primary was just creating that string. The secondary was either the helical or the pleated type of folding. The tertiary was how that protein then folds again on itself. And the quaternary, if that's involved, is 2 polypeptides coming together. So some of the folding can be done spontaneously based on the charges within the protein. It'll just sort of do some folding. But some of the other folding needs to be done by these chaperone proteins that help to form the protein, keep it from folding, misfolding, and creating something that the body can't use. There are other kind of chaperone proteins that help to refold proteins. They're called heat shock proteins. Now, when we go into a sauna, for instance, if we're in there for a while, we can start triggering the release of these heat shock proteins. And these heat shock proteins are pretty amazing. They can take proteins that are misfolded or damaged and repurpose them, refold them properly so they can go back and do their jobs. So getting into a sauna can help refold misfolded proteins within us. So it's kind of a good thing. Going just a little bit further down the rabbit hole. So there are 2 locations for ribosomes. Ribosomes can be within the cytoplasm, within the cell, within the cytosol, within the cell, as free ribosomes, or they can be on another organelle called the rough endoplasmic reticulum. The reason why it's called rough endoplasmic reticulum is because there are ribosomes studded all over the outside of it. And the difference between the locations of those 2 is that the ribosomes that are just free floating within the cytoplasm create proteins that are used by the cell. Those that are created by the ribosomes that are on the rough endoplasmic reticulum are used outside of the cell, or for the cell membrane, or for these things called lysosomes. Lysosomes are these organelles that are filled with acids and enzymes that if there's something that needs to be destroyed, something that doesn't work anymore, it's passed to a lysosome. The lysosome pretty much breaks it apart and then spits it out, and all the things that this thing was formerly made of is then reused. Now, in the beginning, I was talking about some of the things that proteins can contribute to, and I mentioned muscle and things like that. But what's kind of interesting is that bone is also created with protein. In bone, about 50% of its volume or 30% of its mass consists of collagen protein. Kind of wrapping up, there's one little fun fact that I wanted to pass along. You may have noticed, or you may already know, that when you take a steak and you salt it and then put it in the fridge and let it age for a while, the steak actually becomes more tender. And the reason for that is the sodium and the chloride break some of the hydrogen bonds in those folded proteins. And so those folded proteins start to relax, and that's called denaturing. So once those those proteins are denatured, they become a lot more tender. That's the macronutrient protein. I think I went off some pretty deep portions of this, but I wanted to sort of give an idea of not only the beginning of the creation of amino acids and proteins on our planet, but also how proteins are created in our cells. These things, these proteins, are so incredibly complicated, and they do so many things. They're so important. Hopefully this gives a really good rundown on just a basic skim over of all the things that proteins are made of, how they function, what they do, where they came from, and hopefully this is clear enough for you to go off and do some more research on your own to find out more about things that are related to protein. So thank you once again, and please, if you like this, like it and subscribe, and I will see you next time.